And disorders, we have the Down syndrome disease, which is a trisomy 21, where you have 3 things like this. Next, we have the Turner syndrome, when you're missing one X chromosome. So when you produce a child that is usually a female, because 2 X's indicate female, and if you do not have a Y, that means you are female. And we can see that there are an X here, but we're missing the other one, which turns into a Turner syndrome. Klinefelter's, and we have when someone has 2 X's but has 1 Y, which means that the Y was placed there, but there is supposed to be either male or female. We have an extra of either. Then Edwards syndrome is trisomy 18. Now these are the types of mutations in genes. Mutations. A gene mutation is a permanent in the DNA sequence that makes up a gene. So if we have the original sequence TGGCAG and we're going to use substitution for it, the mutated sequence would be TGGTAC. Instead of a C, it turns into a T. Now for insertion, the original sequence would be TGGCAG, but insertion is when you insert a codon to it. What would be TGGTACCAG, creating an imbalanced number. Deletion is when you delete a set of codons or just letters. Deletion would be when we have the original TGGCAG, but if they were to delete C and A, it would only come to TGGG, which is very dangerous. Next, we're going to talk about reproduction. The difference between sexual versus asexual. Sexual are the patterns of reproduce that involves their production and fusion of a haploid sex cells, haploid sperm from father's fertilized haploid egg from mother that develops into a multicellular organism through mitosis, results in genetic variation in multicellular organisms, good for changing environments, slow process, produces low number of offsprings, meiosis equals formation of sex cells. Also, sexual can mean there are 2 parents, male and female, male and parent, well that's supposed to be female, and combined set of genes different from parents and siblings. Examples of that are chickens, iguanas, lobsters, sharks, humans, and butterflies. Next we have asexual. It means where it's one parent and copies parent genes and their carbon copy or exactly the same as their parent, and there are no significant crossovers in the gene mutation at all, and it's all basically the same thing. Bacteria produce asexually via simpler process known as binary fusion. Examples of that are strawberry, euglena, and yeast. Now, talking about meiosis. Now, this might look very hectic to you, but it's going to be helpful. Meiosis is a type of cell division that results in 4 daughter cells, each within half of the number of cells chromosomes of the parent cells. So, we have different phases. We have prophase 1, metaphase 1, anaphase 1, and telophase 1. I'm going to explain prophase 1. Prophase 1 is the chromosomes condense and the nuclear envelope breaks down, crossover occurs, which is right usually there. Metaphase 1, pairs of homozygous chromosomes move to the middle of the cell by the spindle fibers attaching to the middle of the chromosomes, and they would form into a line. Anaphase 1, homozygous chromosomes move to the opposite side of the cell, pulling each of the chromosomes with them. Telophase 1 is the chromosomes gather at the poles of the cell. The cytoplasm divides. And this is the first phase. Next we have prophase 2. Prophase 2. a new spindle fiber forms around the chromosome cells. Remember, in telophase 1, we said that there was a splitting of each of the cells into two. That's why in prophase 2, metaphase 2, anaphase 2, and the rest, you see two cells. We see the spindle fiber forms around the chromosomes, where it forms around them. And in metaphase 2, chromosomes line up at the equator, same thing as metaphase 1. Next is anaphase 2, where the centromeres divide, the chromatids move to the opposite poles of the cells, carrying each them a set of chromosomes. Then we have telophase 2 and cytokinesis. It is when a nuclear envelope forms around each set of chromosomes. The cytoplasm divides in half, creating 4 identical haploid cells. And now we're going to talk about fertilization and how sperms and eggs are created. So we have the spermatogenesis, the oogenesis. Spermatogenesis is it's the creation of 4 sperm cells. It is following the same process of meiosis, but it creates sperms. The production or development of mature sperms. Oogenesis is the creation of eggs. It is when one viable egg and 3 polar bodies. So it goes through the same process as meiosis, but instead of having 4 current eggs, we're going to have only 1 viable eggs and 3 polar bodies. So it starts off like this, and it's going to progressively have one bigger, where each of the chromosomes kind of shifts over. This is when the production of egg cells occur. And then we are done. Yay! This was created by Stephanie and Gabby, and this section was created by Stephanie.